Dilantha Fernando. I'm acting Associate Dean of Research in the Faculty of Agricultural and Food Sciences. I'm really happy with this great turnout. Thank you all for coming. This year we do things a little bit differently in our faculty seminars, and in that we want to focus on partnerships and collaborations. And I think the team that's standing here is a great start. We have 2 people from the Faculty of Science and 1 person from the Faculty of Agricultural and Food Sciences. So Doug is going to, I'm just looking for Doug. Doug is there. Doug Kitani is going to introduce each of the speakers separately. And I'm going to introduce Doug. And I all want you to know one very important thing about Doug, and that he did his PhD in the country that I was born in, and that's the Netherlands. And his PhD was at the Institute of Wageningen, and in the Dutch way how to pronounce that is Wageningen. So I think that's probably the only and most important thing that you have to know.
And never say Vincent van Gogh. In the Netherlands, they will ridicule you or just ignore you. Okay, it's my pleasure to introduce our 3 speakers for today. I will introduce them in order as they're written on the screen there. I'm going to read their bios. Dr. Dilantha Fernando is the Dean of Studies at St. Paul's College here at the University of Manitoba, plus a professor in the Department of Plant Science in this faculty. He works mainly in developing sustainable disease resistance management strategies to reduce crop pathogens in canola and in also cereal crops. He is a keynote or invited speaker at many international conferences. He has been internationally recognized with awards for research excellence in research, teaching, and service. He has 135 peer-reviewed papers and trained over 75 graduate students, many of which have come back today and are in this room. Next is Teresa De Kievet. She's a professor and associate.
You said that wrong, and that's Dutch. Very ferocious.
Oh, well, yeah. Yeah, I
It's
put your
totally
names in
fine.
many languages. It's not just
Okay.
She's professor and associate head of graduate affairs in the Department of Microbiology. She's received her BSc and PhD from Guelph in Southern Ontario, after which she completed a 4-year postdoc fellowship at the University of Rochester. She joined microbiology in 2001. Her research focuses on bacterial biocontrol agents to combat fungal diseases of canola. She's also exploring how these beneficial bacteria interact with grazing predators such as nematodes and protozoa. And finally, our 3rd speaker today will be Dr. Mark Belmonte. He's an associate professor and associate head in biological sciences at the University of Manitoba. So we've got 2 departments represented from science as well. That's good. He received his MSc, BSc, and MSc from the University of Calgary, and his PhD from the Department of Plant Science, actually, here at the University of Manitoba. Oh yeah, under Dr. Claudio Scasola. He then did a postdoc at UC Davis and came back to Manitoba to take up his present position. Okay, his research group uses cutting-edge next-generation molecular and plant laser microdissection techniques to improve crop protection and protect some of Canada's most important agro crops like canola. He has over 50 publications in top academic journals, 2 patents, and numerous invitations as well to present his research findings. He's a recipient of many he has been the recipient of numerous awards and is devoted to promoting science education and research at extension events in Canada, and takes pride in training the next generation of scientists.
Okay. Thank you, Doug. Good afternoon, everyone. It's a pleasure to make this joint presentation as the first joint presentation of the Faculty of Agricultural and Food Sciences. Already, I'm a little nervous to see such a distinguished crowd. We have our President Emeritus, Dr. Emeka Satmari, right here, Dr. Chris Adams, the Rector of St. Paul's College, and many distinguished guests, CEOs I can see from industry and managers from Manitoba Agriculture, and many others, and including lots of heads of department. Normally I speak to plant pathologists, and it's easy. They talk the same language, and it's easier. So if by chance you get bored, please get on with your work, or whatever you are doing, because this is about diseases. It's not great about things that, but what we are trying to show you is how cutting-edge research and the knowledge base that we bring from a student and postdoctoral standpoint can transform agriculture and what it can be in the future. So that's what we are hoping to do today. So hopefully you'll be entertained with some of the things that we are doing. So with that, again, I want to thank my collaborators. Terry has been a collaborator with me for almost, I think, 15 years. So that shows my age and a little bit of her age. And also, the new kid on the block is Dr. Mark Belmonte. He joined us, I think, in 2009 in this collaboration. And so it has been a wonderful collaboration, working on both two main diseases of canola, the blackleg and sclerotinia. So with that, we have also titled this as Dissecting Designer Genes, because throughout the presentation, you will come across how we have been able to dissect out some of these genes that are important in either controlling pathogens or finding novel methods in the host-pathogen interactions. So that's one of the hallmarks of our collaboration. In different ways, you'll see that it has been achieved. And what I will do is I'll introduce what we are going to talk about, and then Terry will give a presentation on some of the work that she's doing in collaboration with my lab, and then followed by Mark. And then I'll come back and finish up with some of the work my lab has been doing. So canola is nothing new to you. I think Dr. Satmari can relate to this very well. She has been very passionate about the canola industry and the canola development. She has asked many questions from me over the years. And it is now a $27 billion crop. And the father of canola, we can be proud, is Dr. Baldur Stephenson, who was from the Department of Plant Science. Now, when it's a large crop, when it has a huge impact on the industry, the economy of Canada, and also on the health aspects like what it is, canola stands at the very top with considered to be the healthiest oil from the point of the saturates. It has only 7% of the saturate component, and there are many monounsaturates in it as well that are considered to be the healthy part of it. So with that, there's a need for us to protect this crop. And that's part of what we are going to be talking about. Now, there are two major diseases in Canada and across the world that has a huge impact on the canola production. Canada stands as the #1 country that produces canola, but there are many other countries that produce canola as well. So the pathogen that I'm going to talk about that is very near and dear to my heart is the blackleg pathogen. It's an ascomycete for the fungal pathologists who are here. And it is a pathogen that can cause disease on the leaves, the stems, and particularly on the area of the stem. Now, Paula, my technician, can relate to how much of cutting that she has done over the years and how strong her hands are during the summer because of the work that we do to understand and identify the resistance that we have in our canola cultivars that we test for all different companies and the industry. So this slide I wanted to put just to let you know, unlike other diseases, canola the blackleg in canola can cause the disease on all parts of the plant. So this is fairly unique because if you take Fusarium head blight, some of you are working on Fusarium head blight, it attacks only the on the head of the wheat plant, whereas the blackleg pathogen can infect everything. Now, I won't get into a lot of details here, although I want to emphasize that we have a trade issue. Thank you, Doug. A trade issue with China on the blackleg. They don't have blackleg, we have. The trade issue itself has cost us about $3 billion each year. So that's a huge part of what we have done in our lab to mitigate that and to start the process of canola movement back to China. Now there's about 70 to 75% of the canola going back to China after the work that we had done and presented to the Canadian government, which is phenomenal when you think about the economic value. And also wanted to mention there is still the problem of blackleg in Canada. How do we mitigate this to get that to the 100% so that there's a free flow of the seed going back to China? Now, one of the reasons that we have an issue with canola in blackleg is that the resistance that was there for almost 3 decades is breaking down. So this I will touch base in my second part of my presentation, so I won't get into details, but the resistance has been breaking down for the last 8 to 10 years. Now, on the Sclerotinia front, one of the key factors that you will encounter is we do not have resistance. We do not have good resistance in the material or the varieties that are bred and released to the industry. And also, the fungicides that are used on the pathogen, we have seen that there's an increased amount of resistance built up. So that fungicide doesn't act anymore as a fungicide. And then this pathogen produces this sclerotia that can live in the soil for a long, long time. So that, again, adds to the problem. So just to finish up my first part of the presentation, to show you some of the disease symptoms on the on canola caused by Sclerotinia. And you can see this kind of shredding, and the pith of the stem is all gone. This is all due to the Sclerotinia infection. If you have such an amount of infection, definitely you can see that there wouldn't be any yield that you can get out of that. There won't be pod formation. So with that, I will now hand it over to Terry to continue.
All right. So as Dilantha said, we've had a long-standing relationship, which has been great for the last 15 years. And so to answer the question then, are there alternatives to controlling Sclerotinia in canola? As you can imagine, the answer is yes. And so something that we work on collaboratively is the idea of biological control. And so I'm sure this is not new to a lot of people in plant science, but that's generally defined as the use of one organism to control the growth of another organism. And there are two main groups of bacteria that have received a lot of attention as biocontrol agents. There's gram-positive bacilli and there's gram-negative pseudomonads. And it's the pseudomonads that we work with in our two labs primarily. And they're grouped into being fluorescent pseudomonads, and there's really three main ways that the biocontrol is achieved. This is the most important for many biocontrol agents, and that's the production of antifungal products. So there's direct inhibition as the pathogen and the biocontrol bacteria meet each other, and that's done through antibiotics as well as lytic enzymes that break down the cell wall of the fungus, and they often work in concert together. There's also induced systemic resistance, so just the presence of the bacteria on the plant can stimulate the plant immune response and make it better able to ward off pathogen attack, and also competition for resources. And the bacteria that we work with pretty much do all of these, but I'm going to be focusing on the antifungal products for the most part. Okay, so the organism that we work with, or that I'm going to be sharing about today, is Pseudomonas chlorophyus strain PA23. We have another Pseudomonas isolate as well called DF41, but in the interest of time, we're just going to talk about this one. And it was isolated by Dilantha's group from the rhizosphere of the soybean plants, and it showed excellent biocontrol against the two pathogens that Dilantha just introduced you to, so Sclerotinia and Leptospirea maculans. And this illustrates some assays that were done in the greenhouse where we've got canola plants that were treated either with a saline control. I don't know if that's anyway working, but that's okay. Sorry, I'll just see if I can figure this out without screwing this up. Oopsie, without shifting this. Okay, yeah, I think I'll use the two. Okay. Saline control, or PA23, and then 24 hours later, they were challenged with ascospores of Sclerotinia. And so over the course of the next 14 to 21 days, we monitor for disease, and you can see that these plants have succumbed to Sclerotinia infection, whereas the PA23 plants are healthy. And so that work was done by Dilantha, and so when we kind of got together and had a meeting, we're both mutually interested in pseudomonads and regulation, that sort of thing. What wasn't known and what we brought to the table was the ability to look at a molecular level at what the key factors were involved in biological control. So what are the compounds being produced and the genes responsible for those compounds? And so in terms of general categories of things that usually are involved in biocontrol, we've got siderophores, and so they have the ability to scavenge metal iron or ions like metal, and that deprives the pathogen of iron, limiting growth. They also produce lytic enzymes like lipases, protease, and chitinase, and antibiotics. And so I've got a whole bunch shown here, and a typical feature of biocontrol bacteria is that they produce a suite of different compounds, even multiple antibiotics that can be broad spectrum. So in terms of PA23, it produces phenazine, so two different derivatives of phenazine, a little bit different one than this shown here, pyrrolnitrin and hydrogen cyanide, and I'll be talking about all three of these today. And it also produces all of these antibiotics, and it produces siderophores as well. So we started with not knowing really too much about PA23 and how it was mediating biocontrol. And so we started with a wide-scale transposon mutagenesis experiment. So my grad students, my little minions, had to go about and test 6,000 mutants. We made a big mutant library, and we tested each one. We tested each one in these antifungal plate assays that I'm going to show you. And essentially, the interesting mutants all came out into two different clusters. Ones that were for the actual products themselves, the biosynthetic genes, were hit by a transposon or interrupted, and other interesting mutants fell into the category of regulators of expression of those products or genes. Okay, so these are the standard plate assays that we do, and I'm showing you a couple of the first interesting mutants that we fished out. So here we have PA23 on the plate, and we've got a fungal plug that gets put in the middle, and it grows out, the mycelia migrate out radially towards the edge of the plate, and if the bacterium is producing something that inhibits the growth, we get this nice zone of inhibition. And you'll notice that PA23 has this gorgeous orange color to it, and that's because the antibiotic phenazine imparts orange pigmentation. So here we have an interesting mutant that actually had an increased zone of inhibition, and it was white. So that suggested it wasn't producing phenazine. And sure enough, when we went in and sequenced the site of transposon insertion, it was in the phenazine operon in the Phys-E gene. So this doesn't produce any phenazine, but clearly it still has biocontrol and actually enhanced biocontrol. And I told you earlier that this strain also produces pyrrolnitrin. And so when we've characterized this strain, what we learned was that it produces twice as much pyrrolnitrin as the wild type, which suggests then that phenazines aren't as important and pyrrolnitrin is important for biocontrol. Okay, and this is a regulatory mutant called GAC-S. It's part of a two-component signal transduction system. This is the GAC-S mutant here, and this is the mutant complemented with the missing gene, and you can see it's white as well, but now the fungus has grown right over top of it. So we've totally lost biocontrol, and it's restored with the complementation. So this mutant bank that we isolated was really instrumental in helping us get a handle on some of the products and the regulatory pathways overseeing their expression. So we had the hypothesis that pyrrolynitrin was the key antibiotic for biocontrol. And so we had to go in and do a targeted mutant of this, and you can see there's a reduction in the zone of inhibition. It's still orange, so it's producing phenazine. It's got some residual antifungal activity, so clearly the remaining products are contributing. If we knock both antibiotics out, we have a total loss of biocontrol, and there's our Phys mutant. So why do we care about knowing what antibiotic is important? Well, now we can really focus our efforts more on pyrrolynitrin, because it was a bit surprising actually that the phenazine wasn't as important, because for many biocontrol pseudomonads, phenazine is the key antibiotic that mediates the fungal suppression. So we know now that it's really pyrrolynitrin. And conditions that favor expression of one antibiotic are often distinct from what favor another. And sure enough, different pH optima favors pyrrolynitrin over phenazine, and different carbon sources. So now we can really focus more on pyrrolynitrin, because we know that's the critical one for biocontrol. And all I want to do here is just show that this extends to the greenhouse, our plant studies. And so if we look at disease severity, just essentially by measuring the size of the lesion on the stems, which is the critical part that damages the plants, we see that with PA23 and the Phys mutant, they show excellent biocontrol or disease mitigation compared to our disease control. The pyrrolynitrin mutant's kind of in the middle, and when you take out both antibiotics, we lose any kind of disease control. And so actually it's great, because these assays really do reproduce very well in the greenhouse. So we get a sneak peek with very simple assays on plates, and it pretty much is mimicked by what we see in the greenhouse. Okay, so one of my loves is regulatory cascades. So puzzles, I always say, if you love puzzles, then you'll like working in my lab, because it's very complicated, often networks of regulation. And so what we've done is we're interested, okay, so what are the regulators that are controlling expression of pyrrolynitrin, phenazine, and hydrogen cyanide? And we've plucked out one by one different suspects that are involved. So we make targeted mutants, and then we phenotypically characterize them. We look at the gene expression, and you'll see that it's a bit of an intricate network. I haven't even got all the regulators on here, but there's back and forth. So there's regulation that exists between the regulators themselves. So it is a bit of a tangled mess at times, but it makes for lots of fun as well too, as we try to put these things together. There's the GAC system I showed you before as a key regulator. And the one I'm going to focus on today for a few minutes is the Phys quorum sensing system. And what quorum sensing systems do is they allow bacteria that employ this regulatory pathway to coordinate gene expression with population density. So if you have a low number of cells, then the quorum sensing system isn't really activated. But as the population increases, this gets turned on, and we get expression of these guys I'm going to show you. So just to take a look at this a bit closer, the quorum sensing system in PA23, we've called it the Phys system. There's two genes that make it up, Phys-I and Phys-R. And the way that these work and coordinate population density with turning on genes is that the Phys-I gene encodes an enzyme called the autoinducer synthase, and this generates autoinducer. And this gene is always on at a very low basal level. So when you have a low number of cells, you get a low amount of autoinducer. But as the population increases in a small contained environment, so does the level of autoinducer. And it then, at a threshold concentration, is able to bind to a cognate transcriptional activator called Phys-R. So Phys-R just functions when it's together with its partner autoinducer, which is C6HSL. So as soon as it complexes with the autoinducer, it changes conformation, and it can now bind upstream of target genes. And I have little black boxes here, which we call Phys boxes. So these are discrete binding regions that are recognized by Phys-R and the AHL together. So they bind, and then they allow transcription through interacting with RNA polymerase of the target. So the Phys-I gene is auto-induced, so as soon as the system starts kicking in, it really, really ramps up, and the Phys operon as well is controlled. So we were interested then, is biocontrol under quorum sensing control? And yes, it is. So here's our wild type, and here's a quorum sensing mutant, and you can see it's white, as we would expect, because the phenazine operon is not expressed, so we don't get that gorgeous orange color. And there's no antifungal activity. And when we've gone in and characterized the mutants, we see that there's no phenazine, there's no pyrrolynitrin, no hydrogen cyanide. There's also no protease or chitinase. I didn't add all those. And it's also reduced in biofilm formation. So this is a biofilm forming bacterium. So this gives us another key piece of information because we obviously need to have sufficient population present in the environment for these cells to function as biocontrol agents. So if the population is insufficient, that they can't be quorate and coordinate and upregulate the antibiotics, then they're probably not going to be really effective biocontrol agents. So the next thing that we did is to look at the global impact of quorum sensing. So we knew that those targets like antibiotics and enzymes were shut down, but this was done with a really excellent collaboration now with Mark's group, because he is our guru on next generation sequencing and RNA sequencing. And so with the help from Mark and students in his lab, they were able to train my master's student, Nidhi Shah, on the nuances of making RNA sequencing, or libraries for RNA sequencing, and all the downstream processing. So that was really a very powerful collaboration, and I'm indebted to Mark from the bottom of my heart for all of the assistance he gave, because to this day I really can barely figure that out myself. So anyway, but what we found is that the quorum sensing regulon actually is quite extensive. So it represents about, I'll just step back and say too, we looked at 2 mutants. So we took out the FISR, the transcriptional activator, and we made a strain AHL deficient, so signal deficient. And what we see is that quite a bit of the chromosome, ranging from 5.5 to 7%, is under quorum sensing control, so quite significant. And I'm not going to belabor this, but all I wanted to say is this was very gratifying because a lot of these genes matched up to what we saw already. Like phenazine genes were down, the pyrrolnitrin genes were down, so we're like, okay, this is a good thing. We've got a boatload of genes, and it appears that they're matching up to the characteristics, which validates the rest of the data for us. And we also did look to see for those fizz boxes that I showed you before, so are they present in a lot of the promoters that are under quorum sensing control? And in fact, it's only a small cohort. 25 of the genes had the fizz boxes, suggesting that a lot of the regulation is happening indirectly, like that cascade that I was showing you. So if one of those regulators is quorum sensing controlled, and then there's a number of genes under that, it's through regulation indirectly of that intermediary regulator. And sure enough, when we looked at our genes, we saw that there were 48 regulators of different families and origins that are quorum sensing controlled, suggesting that a lot of regulation in PA23 in the quorum sensing regulon is indirectly mediated. And this is the last little bit of my story. It's a question that we've been asking lately, is why is PA23 producing the compounds in the first place? Because it doesn't really benefit it that much just for the purposes of fungal suppression. And others before us have come up with this idea that it may be to ward off grazing predators, because this is very energetically costly for the cells to be producing these compounds, so it must impart a fitness advantage, otherwise they would stop making them. And so I don't know if this, so we essentially have been using the model organism Canorhabdus elegans, which is a nematode that feeds on bacteria. So let's see, I don't know how I'll get this, just the mouse? Okay, thank you. This is just, you know, when you get a little movie, you have to be very cool. So here's the Canorhabdus going through. So this is basically what this is it's a lawn of bacteria. So the nematodes feed on the bacteria. So we can grow our PA23 and our different mutants as a lawn on a Petri plate, and then put the Canorhabdus elegans on the plate, and they will feed on it, and we can determine whether there's any toxic effects. So if they are happily eating away, they go through their developmental stages, they get fat, they lay eggs, or if it's toxic, they start to die, basically. So I'm not going to get too much into this. We've got a suite of different regulatory mutants and individual biosynthetic mutants, and the take-home is we do these survival assays. So my poor grad student, Manman, that I showed you, sits, and every 2 hours or 1 hour, I guess, is looking at this chart, goes along and pokes the nematodes and sees if they're still moving. And if they're not, then you get a little X with in their little eyes or whatever, not really, but a little score sheet. I don't know how she does it. Anyway, and we come up with these survivability curves. And the take home here is that we've discovered that pyrrolnitrin and hydrogen cyanide are toxic to Canorhabdus elegans. And the other thing that she has done is something called a binary choice assay. So we put one type of bacteria on one side of the plate, like the GAC-S mutant, for instance, that has no biocontrol products, and the wild type, and you put the worms down the middle, and you see which side that they migrate to, because sometimes bacteria produce attractants or repellents. And so what we see is that for the most part, so this would be the worms at the starting point, and then you count them afterwards. It's mind-numbingly exciting work that goes on in my lab between picking colonies or counting worms or whatever, but you do get exciting results, and it's great to be the prof. So, but the bottom line too here is that both hydrogen cyanide and pyrrolnitrin act as repellents. So take home, we think this is very good in terms of persistence of PA23, because if you put a biocontrol bacterium into the environment, they need to be able to colonize and they need to be able to persist. And so if they're able to repel predators that might be gobbling them up, that means that hopefully they're going to survive for longer periods. We haven't really extended this too much to soil. This has all been done in vitro, but it's looking good for the early stages of things. So to wrap up my section, where are we with what we've learned? Well, PA23 produces a wide range of different compounds. In terms of antifungal activity, pyrrolnitrin is definitely the key one. And it's governed, the expression of all of these compounds, by a very complex regulatory hierarchy. Included among that is the quorum sensing system, which oversees biocontrol and about 7% of the genome. And we're excited that PA23 exhibits both nematocidal and repellent activities, primarily through those two products. We haven't looked at it, but it's a common question. We're thinking about it. But there are a lot of nematodes that represent threats to crops, including canola. Okay, so it's potential that PA23 would be able to have nematocidal activity against them. And my PhD student Akram has just started, or the last couple of years has been looking at protozoa and the model organism Acanthamoeba, because protozoa also graze on bacteria and change the microbial community structure. And we're seeing pretty much the same thing, that hydrogen cyanide and pyrrolnitrin are very important for that. And with that, I'm going to hand it over to my excellent collaborator, the third one in the dog and pony show, Mark.
All right, thanks, Terry. Thanks, Dilantha, as well as thanks to the Faculty of Agricultural and Food Sciences for extending such a warm invitation, especially for me to come back onto what I consider to be home soil, onto my alma mater, and talk a little bit about some of the work that we've been able to achieve together through a collaboration. And I'm going to switch gears a little bit. So I'm going to switch gears away from sclerotinia as well as PA23 and go back to some of the work that we're doing in trying to find new innovative ways of protecting canola against the devastating fungal disease blackleg using genomic tools. So one of the things that we've encountered over the past few years is that blackleg is definitely one of the most important problems that's affecting canola growers across Western Canada and around the globe. And so while we've started to identify some of the key resistance genes that's responsible for protecting this crop, we still don't know all of the genes that's responsible for defense. So identifying some novel genes, identifying some of these novel regulators that can help to control plant immunity as well as plant defense can have really great benefit to canola growers across Canada. So one of the things that we decided to do is we decided to use our knowledge of RNA sequencing and bioinformatics in order to profile all of the genes and gene regulatory networks that are found within a universally susceptible line, as well as a commercially available resistant line. So we were working a lot with some of Dilantha's collaborators, and what we did is that we used the susceptible line Westar. So a lot of us are familiar with this. It's universally susceptible against a number of pathogens as well as diseases. And then we were also lucky enough to access a resistant cultivar. And that resistant cultivar I'm going to label here as DF78, which carries the LEPR gene. So one of the first questions that we had was, how does disease manifest itself and what does it look like between the two lines? So what we have here is the canola cotyledons. So these are the first embryonic leaves of the seed. And as Dilantha mentioned before, blackleg really attacks all stages of the canola life cycle. So what we did is that we inoculated the canola cotyledons in the Westar, or the susceptible line, as well as in the resistant line. And what we can see that within a week, there's no real phenotypic difference between the two lines. But only 4 days later, at day 11, we can see in the universally susceptible line that the cotyledons are completely diseased. So the lesions have pretty much taken over the entire cotyledon, compared to the resistant line, where we can see nice levels of resistance. So we don't see any disease within this cotyledon. We can then go ahead and quantify lesion size over time. So what we're looking at is before, during, and after the infection process. So we don't really see, again, any difference for the first 7 days after inoculation. But it's this 7 days which is considered to be this critical point. And after 7 days is where we start to see huge differences in the way that the plants are responding to this disease, where we see a dramatic and significant and profound increase in lesion size in the susceptible line compared to its resistant counterpart. So as Terry mentioned before, we like to use next generation sequencing technologies in the lab in order to profile all of the genes that are found within the plant. And so what we did is we profiled the genes before, during, and after the infection process, day 3, day 7, as well as day 11. We then made the libraries in-house using a low-cost RNA sequencing protocol that generally costs around $10 per library. We sent off our sequencing libraries to Genome Quebec, and then we get back an absolutely huge amount of data that I'll talk about in the next slide. But what we can see from these cotyledons is that something has to be happening directly at the point of inoculation that's preventing the fungus from spreading in the resistant line. So something has to be happening at the cellular level. In order to address that question, we can use a microscope that I was lucky enough to get in my lab called a laser microdissection scope, where we can literally cut out individual cells and tissues from the leaf, in this case a cotyledon, in order to examine which genes are specifically being activated directly at the site of infection. So this is just a summary table of all the data that we get back. It's not a really big deal what all this data means, but what I wanted to draw your attention to was this middle line right here, and that's the total number of bases that were sequenced. For our experiments, we're sequencing over 74 billion bases. That is an incredible amount of data, or big data, or what my lab likes to call the data apocalypse. So how do we go about managing all of this data, and how do we go about making this data into something that's biologically meaningful? Well, the first thing that we can do is we can compare the genes that are considered to be differentially expressed between tissues that were infected with the pathogen versus those that were treated, for example, with water or a mock inoculated control. We can then create what's called these Venn diagrams, and we can compare the differential gene expression in the resistant line, which is found in this dark blue box, compared to the susceptible line, which is found in this light blue circle. We did this before, during, and after the infection process, day 3, day 7, and day 11, and what we can see are pretty big differences in the way that the plants are responding to infection in both the resistant line and the susceptible line. So early on in the infection, we can see a large number of genes are being activated in the resistant line, again, to promote defense, to promote immunity, and to prevent the fungus from spreading into the plant. By day 7, we can see pretty similar numbers of genes that are differentially expressed between the resistant line and the susceptible line. And then by day 11, this is where the cotyledon is completely destroyed in the susceptible line. Huge numbers of genes are differentially expressed, or what we're like to consider a runaway expression, where the plant no longer is able to control what's happening within itself. So what we've been able to do so far is cut down these 70 billion data points into a few thousand genes. But we wanted to better understand what these genes do. In order to answer that question, we did what's called a gene ontology term enrichment analysis, just to look at some of the biological processes that are considered to be statistically enriched in these data sets. We can then go ahead and plot this data as a heat map, again looking at differential biological processes before, during, and after the infection process. It doesn't really matter what all of these lists of biological processes are, but what we can see in that the resistant line, we see increased defense, increased genes that are involved in immunity, increased genes that are involved in signal transduction cascades that amplifies that defense or immune response. And we also see a biological process called callose. In this case, callose is specifically upregulated or specifically turned on in the resistant line. We can then go ahead and validate these biological processes using histological stains. And what we can see are quite dramatic differences in the way that the plant is able to defend itself in the susceptible line, where callose, or this fluorescent blue stain, is a little more scattered, compared to in the resistant line, where this callose has a tight, nice ring around the point of inoculation. What the callus is doing is it's preventing a physical barrier for the fungus to prevent from advancing. So again, the plant is able to mount a very quick and coordinated defense response at both the cellular level as well as the gene level. So we wanted to break this down even further. We wanted to try and identify some of these key genes. Again, we just looked at differential gene analysis. We looked at the genes that were specifically expressed in the resistant line and compared those to the susceptible line. What we were able to do now is narrow down those tens of thousands of genes into 54 genes. These are 54 genes that were specifically expressed in the resistant line and that were not expressed in the susceptible line, or what we like to call the core 54. So what we're able to do now is we're able to dissect what was 72 billion data points into 54 key regulators. What's really great about plant biology is that we have access to a number of research tools that we can start to investigate some of these genes. What we did is we started to dissect out which genes are the most important for plant defense against blackleg by looking at different mutants that we consider to be susceptible to blackleg. In this case, we're using canola's neighbor, or Arabidopsis, which is considered to be a model plant. What's really interesting about Arabidopsis is that it's considered to be a non-host for blackleg, which means that under normal wild-type conditions, blackleg cannot infect Arabidopsis, which is exactly what we see here. However, if we start knocking out genes that we think are critically important for immunity, for defense against blackleg, we should see some sort of manifestation of disease. And that's exactly what we see here. In a number of these mutants that correspond to receptors, that correspond to transcription factors, that correspond to immune regulators. What we can now do is get a much better understanding of the specific genes that are responsible for blackleg disease and start to integrate them back into canola. So now that I've talked a little bit about some of our broad analysis looking at whole transcriptome of cotyledons, we know that disease is going to be different between the resistant line and the susceptible line, and that something has to be happening directly at the site of infection. So how do we go about accessing those cells? Well, one of the things that we like to do in the lab is use a laser microdissection scope. And what this laser microdissection scope does is it cuts out sections of tissue. In this case, what we're looking at is a cross-section of a cotyledon. So here we have the infection site. Here we have sites adjacent to the infection site. What we have is the epidermis, the mesophyll, as well as the vasculature, the three primary tissue systems of the plant. In this case, we're cutting out a 200 micron section directly adjacent to the infection site, which represents roughly about 2 cells. Because again, we know that something's happening directly at the site of the infection, and that the plant should be able to mount some sort of defense at the cellular level. We can then go ahead and collect literally thousands of these sections in order to get enough genetic material to profile this material either using directed quantitative PCR or next generation RNA sequencing. So what I wanted to do here is just provide you with one example of what we've been able to discover with the power of laser microdissection coupled with quantitative PCR. In this case, we're looking at a marker gene called pathogenesis-related 1, or PR1, which has canonically been associated with plant resistance against bacterial as well as fungal pathogens. What we did is that we cut out 200 micron sections, so again about 2 cells, and migrated away from the point of infection. So we looked 0 to 200, 2 to 400, and 4 to 600 microns away from the site of the infection. And just to give everybody an idea for the micron scale, the average human hair is anywhere between, let's say, 75 and 150 microns. So we're cutting out what's considered to be a human hair, about 3 human hairs in a row, because this is how quickly and how rapidly the plant is able to mount these different responses. So what we did is we compared the expression of PR1 in the susceptible line that was treated with water, the resistant line that was treated with water, and we see no expression, which is expected because PR1 shouldn't be expressed in the presence of water. In the susceptible line, 0 to 2, 2 to 4, 4 to 6, we see a small and gradual increase in the expression of PR1. But in the resistant line, again, the line that was able to mount a very quick defense, we can see an over 1,000-fold increase in genes that are critically important for defense, and that this expression gradually decreases as you go away from the site of infection, or what we like to call the establishment of a plant defense gradient. And we're following up this idea right now in the lab by looking up different types of genes as well as profiling all of the genes that are found within canola at this cellular level. So what I wanted to do today was talk a little bit about how we're using next generation sequencing technologies to better understand host pathogen interactions. We know that large numbers of genes are expressed before, during, and after blackleg infection. We can use modern molecular tools coupled with laser microdissection to be able to understand what's happening directly at the site of infection at the cellular level. And if we can start to identify these genes and characterize these genes in the lab, we hope that we can then contribute to plant resistance, which can help to protect what we like to consider Canada's #1 crop. So what I'd like to do now is pass the microphone back over to Dilantha, who's going to talk a little bit more about his work in blackleg and protecting canola in Canada.
Thank you, Mark. Last lap. So hang on. I'll not take much of your time. So getting right into some of the work that we have been doing in the last couple of years. One of the things that I mentioned earlier was that there was a breakdown in the resistance that we have in canola varieties in Canada. And no one in Canada particularly knew what resistance genes they had, whether it was Monsanto, Bayer, BASF, whoever who was breeding for disease resistance. We did not know what resistance genes they carried. They knew only that it could be resistant or susceptible or moderately resistant or moderately susceptible. So our lab was entrusted, and all the companies joined in a consortium to find what kind of genes were in there against the blackleg pathogen. So this was a really interesting study done by my PhD student, Shuhua Zhang. And what she found was that 65 to 70% of the canola cultivars carried the RLM3 gene. Now, at the same time, one of my master's students, Zachariah Liban, studied the pathogen population out there to see why this breakdown was happening. And what we found was extremely interesting. 97% of the isolates of the AVRLM3 was the virulent isolates. So only a few were avirulent. So bulk was virulent. And if you match the two, you see what was really happening out there in the farmer's field. So the farmer buying seed from, let's say, Monsanto, saying that that was sold as resistant to blackleg, was not exactly, the variety was not performing as it should because of this pathogen population that has shifted from an avirulent standpoint to a virulent population. So with that, we started to see this breakdown and the disease appearing in farmers' field more often than what we would love to see. So as I said, the resistance was labeled just like this in the varieties, and this did not make any sense to the farmer, because when they buy, they're happy, but very soon they will be sad because their crop has failed because of the blackleg disease, because they thought that it was resistant, but in the field it was not performing as resistant. So what we proposed was to move away or have the labels with the seed to be sold with a label of the resistance gene. So if you have the resistance gene, then it is advantage to the farmer, because they can then understand what resistance genes should they be putting into their field if they had had blackleg in the past. So the canola industry, with a lot of discussions and consultations with the canola seed industry, finally came up and introduced this labeling system that we had been promoting for quite a while. And it was it began in 2017. That was the first year. So what we wanted to let the farmers know was that if you put the same gene back to back in 2017 and 18, it would be a bad rotation. Now, most of the farmers would not go canola on canola. They might go canola, wheat, canola. Even with that, the pathogen has survived, and the inoculum of those virulent isolates that I showed is still in the field. And with that, it will be a bad rotation and the crop will fail. So what we were proposing was go back to a different label from this, look for another gene that would not match the pathogen population, and introduce it into the field, and you will have a successful crop. And so with that, we also wanted to give an opportunity for the farmer to decide which variety that they should be growing. To know that, they had to know the pathogen population in the field. So with a collaboration that I had with Dr. Hossein Bohan's lab in Agriculture and Agri-Food Canada, we were able to develop what is known as CASP markers to directly identify the races that are prevalent in the field. Now this is given to different testing labs, including the PSI, the Pest Surveillance Initiative in Manitoba, to identify the pathogen to the races, the exact race that is there in the farmer's field. So when they know that, so let's say they knew it was AVRLM3, the virulent isolate that was present, then they stay away from the RLM3 and grow RLM2 or RLM4. So this is something that I was very happy to see. The Monsanto billboard here explaining how it works and what kind of genes they carry. And this is the old terminology, the way that labeling was done, and now this is the new labeling that is carried. So this is excellent news to the farmer and to the industry. And just a few days ago, I got this slide. If you can just dim the lights for 1 minute. Yeah, so you can see without any difficulty that this side is completely dead and this side is well alive and excellent crop. So this was RLM3 gene that had been grown in 2017, going back, this is 2018, going into 2017 RLM3. So the crop is diseased. But this is in RLM4 going into RLM3, and the plant is extremely healthy. So this is data from New Dale, Manitoba from this year, and I'm really excited to see this. And one thing as a researcher, the researcher's delight is to see data moving to recommendations to be used on the farm. And that's exactly what we have been able to achieve. And I'm really happy that the students and the postdocs who have worked in my lab have contributed to this effort in a big way to change and transform the way that agriculture is going to be done. We are not proposing our gene rotations instead of crop rotation. Crop rotation is still the king. But if farmers are not doing the crop rotation in the proper way, which can happen because of the economics, then our gene rotation will kick in and help the farmer to mitigate the disease and and also help us in the trade issues that we might have. So ensuring the sustainability of canola production, market access to countries like China and other countries that have restrictions on when we have blackleg, and also strengthen the Canadian agriculture and our farmers, and more importantly, be proud of what we had started right from Department of Plant Science at the University of Manitoba. Thank you very much, and I would like to take this opportunity. without the help of many students, including my technician, Paula, most of this cannot happen, and also the funders who have helped all three of us in many ways to do what we have been able to talk about today. Thank you very much.
Thank you very much. I have a pen for each of you so you can write down the questions. So I know it's close to 4:30, but let's take 3 questions, and then for the remaining questions, feel free to talk to the presenters afterwards. Any questions?
First of all, very good presentation. And I have a question for Dr. Fernando. And do you know any reason why China has no black lake?
Very good question. It's a difficult question to answer, not exactly knowing much of the topography and the environment. Now, assuming that the environment is the same, assuming that their cultivation systems are fairly similar, some of the reasons that might be not allowing the pathogen to establish. Now, I'm thinking that there might be inocular, but it may never have had an opportunity to establish. It's something like some of the diseases that we find here. So one of the cultural practices they do and we don't have is going to paddy rice after a canola crop. So paddy is grown under in submerged water, and that might be preventing, and we did a study to see whether the inoculum can survive, and they cannot. And that might be one of the contributing factors. And in the good old days, the farmers and the peasants used the stalk that is left behind after harvest as firewood, and mainly in the rural areas. So that itself takes care of the inoculum that would be staying around. Now, one of the things that has increased in our farming systems is no-till to minimum-till farming. So that increases the ability and the pathogen to establish on stubble and stay and overwinter and come back the next season. And also with tight rotations, it increases that factor. So I'm thinking that those might be some of the reasons that China still doesn't have the black leg. By the way, China grows as much as canola and rapeseed as Canada, so that's part of the reason they are very protective of their crop and not to see the pathogen moving there.
Another question.
I have a question to Dr. Mark. So in your case, can you tell me what the exactly are results the mutant was overcome by black leg?
Can I tell you exactly which ones?
Yeah, which is the mutant one.
It's not that one. So, yeah. So those are quite. We're quite happy to discover that some of those mutants were susceptible, and yep. So some of that information is no longer under an embargo, and so it has been released in a publication that we just put out late last year. And so a couple of those genes are receptors, and we know that the receptors are the first genes and the first molecules that the pathogen encounters, and so we consider those to be really important, and so we're following up and functionally characterizing some of those receptors or first responders in the lab right now.
Any last question?
I'm Patty Roche, my agriculture. This is not my subject area. I was in and out of understanding and nuance, but I think I got it. I'm responsible for the next version of ART. So my question is maybe impossible to answer, but over what time period would all of this work have been done? How long does it take to do this kind of research? And bonus question, how much money would this represent?
First, for my part, the ART-gene rotational studies started in 2011, very slowly because we didn't have funding at the time. It was only a concept and a thought that we had in our lab, and we started that. But from 2013 to 2018, that was the period we had funding. So the funding really helped, and it was about, I would say, close to $500,000. The full spectrum. Now, I presented only a fraction of what we have done to understand this and come to what we have. This was just to make it easy for me to explain, but the total would have been about $500,000.
Yeah, and I would say ours has been progressing since about 2005. So we do have some applied money from group collaborative stuff, but primarily NSERC is funded by research. So I would say probably in the neighborhood of $250,000. So when you're not doing field studies and that sort of thing, a lot of our stuff is lab bench stuff. It's not quite as expensive. So, but when you get into the next generation sequencing, dabbling on that, it starts to get much more expensive. So Mark's numbers are probably going to be an extra order. Yeah.
How much do you got? Yeah, so we've been really lucky, all three of us together, to have received funding through the province in the past, which has contributed to some of these results. We've also been really lucky to receive funding from the Canola Council, the canola growers, and the level of funding really depends on the type of project that we're interested in. I feel that we've done an excellent job trying to reduce the costs, either by doing some biohacking in the lab, but also in recruiting some great students where we can leverage those funds that will also contribute to their training.
The HQP training has been phenomenal in all three programs, and they are now contributing back to the industry in different ways. Most of them are the pathologists or microbiologists working for agriculture and all the seed industries like Monsanto.
Well, thank you very much again, and thank you for being here.